Hello, everyone, and welcome to this interview that we've got here with Guppy from Fred Hutch. So, just to provide some quick context, this interview is done as part of Bingothon Summer 2021. If you've watched the event, you may remember that we've had short snippets shown during the intermissions, showing some key moments of this interview. And now here we are with this full interview that you can watch here if there's anything you were more curious to know about, or if you just want to know more about Fred Hutch in general. So I am Pikastroff, one of the main organizers of Bingothon. I'm also accompanied here by Floa, one of the other main organizers here of Bingothon, and of course, the one and only Guppy. So how about you introduce yourself?
Thanks, Pika. Thanks, Flo. I really appreciate you having me back for a third time. My real name is Amitabha Gupta, but people call me Guppy. I am the Scientific Content Strategist for the Philanthropy Department at Fred Hutch. And I used to be a researcher until 3 years ago. And my job now is to keep an eye on the research and figure out what's going on and help translate it to all of you.
All right. Yeah. Awesome. That's now the third time we're talking here together. For those of you that didn't know, we already had Guppy here with us, like for Bingothon Summer 2020 and Bingothon Winter 2020. And our discussions have gone a long way since then because things have just changed a lot. But, you know, for those of, just in case, for those that may not be familiar with Fred Hutch, would you like to explain a little summary of what Fred Hutch does in normal circumstances?
Yeah, so that's a great question. So Fred Hutch is a cancer research center located in Seattle, Washington. But what we're well known for is pioneering the bone marrow transplant, which is a curative therapy for many blood disorders. And from that, we've got to immunotherapies, which are now being used for a lot of cancers as well. But along the way, while we were discovering that, we realized that people who were being transplanted and saved from their cancer were also dying from infection. And so we recruited a bunch of infectious disease specialists. And at Fred Hutch grew out this group of researchers that studies infections, to that point that we launched a global HIV vaccine trials network out of Fred Hutch, where we test HIV vaccines across the world to try and treat this other disease that's been killing millions. And so we already had about 25% of our faculty researching viruses. And so when this pandemic came, we just switched focus from one virus to another, and it was pretty seamless. And to that end, we've also taken on the job of coordinating the 5 major vaccine trials that are being done by the U.S. government. So basically, the Moderna, the AstraZeneca, Johnson & Johnson, Novavax, and potentially a 5th one for Sanofi, which is to come. And we've been coordinating the trials for that to make sure that the data from one can be used to study compared to the other, so that we can compare apples to apples.
And I was just about to ask you about the summary of the actions Fred Hutch took during the COVID-19 pandemic ever since all that started. But I think that really does pretty much a good summary. You even have open, like if I'm not mistaken, it's the COVID-19 Clinical Research Center.
Yeah.
Even that was more late last year that it was opened up, if I'm not mistaken. Am I correct?
That is absolutely correct. And thank you for bringing that up. Yeah. So, we're seeing across the world right now that there's not enough vaccines to go around. And for certain places in the world, it'll be later this year or maybe even into next year before they get vaccines. There's a lot of people that need to get vaccinated. So along the way, we need to find sustainable treatments that are already approved that we can get out to people very quickly once they get COVID, so we can reduce the severity of illness for all the people that are getting infected and dying. I mean, I'm from India, and looking at the news every day is really heartbreaking for me. And if we could only have like some treatments that we could ship over there in bulk that are already approved, that could save so many lives.
Yeah, I remember we've talked in previous interviews about also, the importance of, not just the vaccines, but also potential treatments
Right.
that could also be vital. And as you just said, especially with what's happening right now in India, at least at the moment of recording this interview, this would be the kind of thing where further research into that is definitely useful. Because I can read here indeed that the Clinical Research Center is doing work on not just the vaccines but treatments, and so perhaps could you talk actually about where that research is going regarding like treatments as well for COVID-19?
Oh, absolutely. So there's a few treatments that are, so I believe some of you may remember last year, the then President Trump got ill with COVID-19, and he received some treatments which were called monoclonal antibodies. So antibodies are these Y-shaped molecules in your body that recognize foreign invaders, so bacteria and viruses, attach to them and target the rest of the immune system to come degrade them. So we are testing a bunch of those antibodies that are targeted to the spike protein. So these are treatments that basically your body may not have antibodies yet to fight the virus, but we will give you the antibodies you need that can destroy them. So there's a couple of those treatments that are underway. There's a couple of antivirals. So viruses have a life cycle. They enter the cell through a certain way. They'll make copies of themselves. But the way this virus copies itself, we could potentially treat that with drugs that already exist, because the virus needs to do something to make copies of itself. If we can block that, we can reduce the severity of illness. And then there's other things that include treatments that can reduce how severe the immune reaction is. A lot of people are dying because the immune reaction is too severe. So if we can give this treatment to sort of damp it down just a little bit, that can also potentially save a lot of lives. So there's all these different drugs that are currently in trials, early in treatment. So when people just get sick, that we can give it to them and make COVID into your seasonal flu or cold.
So, and maybe to ask, so how far are we into these trials so that these methods could become eventually publicly available for anyone?
Absolutely. So there's a couple of these trials that have come to the point where they're going up for approval, but the more encouraging ones are there was a recent publication about a drug called tocilizumab or toci, which can reduce death from COVID-19 by dampening the immune system. That's already approved. So that can, that research has been done. Now we can say, all right, this is an approved drug. You can use it. We're providing best practices. So there are some treatments that haven't been approved that are moving along the pipeline, but there are treatments that have already been approved for other illnesses that can now just be repurposed. And hopefully that will start making an impact sooner.
I see. Yeah. Indeed, once those treatments really can get advanced, like further along the pipeline, I'm really curious to see how that will impact things. I'm just really wondering how different it will be maybe between like first world countries and third world countries. Like once again, I'm thinking in the case of India, because it's such a tricky case. Something that's a lot in the conversations lately is variants of the virus. So actually, just before we get into this discussion about India and the variants and the other variants as well, could you do a brief summary of how variants work in general?
That's a great
COVID?
question. So all viruses make copies of themselves once they enter our bodies. And the way this virus is made, every time it copies itself, it makes some mistakes. And sometimes those mistakes are bad for the virus and those viruses die away. But sometimes those mistakes provide that particular virus some sort of competitive edge over the predecessor. And so either it can enter cells more easily, or it can escape the immune system, or it can be transmitted more easily. And so you start seeing these. So those are. The errors are called mutations, and soon you start seeing like a virus that has a mutation from here, and then it picks up a second, then it picks up a third, and that's what's happened with all the variants we've heard about. They've picked up numerous mutations in the spike protein, particularly. So spike protein is the part of the virus that it uses like the key to pick the lock to our cells and then enter our cells. And so all these variants have a ton of mutations in the spike protein that are giving it some sort of competitive advantage. The big one is the B117, the variant that has been ripping through the UK and then Europe. Those changes to the spike protein allow it to be more infectious. And by that I mean, say one person walks into a bar with the original COVID and another person walks in with B117. This person will infect maybe 10 people. The person with the B117 will infect 15 people. And so it's just leading to more spread of the virus in one way or the other, possibly due to the fact that it makes more copies of itself. And so there's more virus that a person with B117 would shed in a public space versus the person without.
I see. Yeah, increasing even more the risk of spreading, how to make the problem even worse, basically.
Yes.
And yeah, that's why then it justifies really those kind of fears. Because once the word variant kind of appears like in the media, it really creates this kind of fear that you have. And considering that indeed it can impact how it spreads, in that sense, it seems justified. But of course, I remember something we also talked about, like in our previous interview, like in winter 2020, was how for the most part we don't really have that much worries about vaccines and potential variants. If I recall correctly, they should still be effective, against different variations, because of that that spike protein. If I recall correctly, at least. Now, of course, because it's in the headlines everywhere, people are asking that question of, well, are vaccines still likely to be effective? Obviously, I suppose you can't really provide a one-size-fits-all answer, because by definition, I guess they're all kind of different. But do you have, compared to what we said like back in winter 2020, like do you have a different stance, or what would you have to say about it?
So I'll say this much to start with, right? Like, there have been variants since the start of the pandemic. There have been many different variants that existed. They didn't really have a competitive advantage or disadvantage over the others. The reason there are variants, there's hundreds of millions of variants. When they become variants of concern or variants of interest, then we should start focusing on them. And what I mean by that, they become variants of concern or variants of interest if you see a spike in a country and an increase in cases that is associated with one variant, or you start seeing more deaths and you see a certain variant show up in that country. Otherwise, there's a variant, it comes, it goes, it doesn't matter. And yes, it is really crucial. The scary part about these variants is that the mutations occur in the spike protein, which is what a lot of the vaccines are targeting. And so for a lot of at least for Pfizer and Moderna, we don't truly know how they'll do against variants, because when they were tested, we weren't talking about variants. They were tested summer of last year, and there was no variant talk then. AstraZeneca, Johnson & Johnson, Novavax, we do have a little bit more data because they have been in the field being tested against the variants when they were out there. So we have a little bit more data on that. And what we do know is that by and large, the Johnson & Johnson and the Novavax are pretty good against other variants too. And so that's a plus. AstraZeneca is not as good against the B1351, which has been circulating in South Africa, but it's good against the others as well. So some vaccines are able to fight off all of these, but I will also say that we have real world data, right? So the UK, the US, and Israel have taken on really robust vaccination programs. And in all of those countries, B117 has been the dominant strain. And in all of those countries, we're now starting to see a reduction in cases. So that suggests that the vaccine is really good against B117. So we should be very hopeful here.
So with recent developments, we see more and more countries banning or just renewing their opinions on certain vaccines. And for example, there are some people who already got the first vaccine shot, but now they can't get the next one with the same vaccine. And in how far is that possible in mixing basically these vaccines together? Is the effect still the same, or do we have a reduced effect with this?
So that's a great question. I think the answer is that we don't know. We haven't been able to test this mix and match strategy. It's possible that it would be good because there's this thing in the vaccine world that's called like heterologous prime boost, where occasionally if you give one type of vaccine and then do another, you can actually see an effect that is over and above both of them. But those have been done in like properly tested scenarios and proper, everybody's been tested, everybody's part of a trial, we've made sure everything's okay. That is not the case here. I think the case here is that we don't actually know whether you can mix and match. And so when we don't, in the absence of knowledge, I would definitely recommend sticking to trying to get the second dose of whatever you got from the first dose. And for things like AstraZeneca, actually, when the UK did the first dose and the delayed second dose, they even found that a delayed second dose even helps the efficiency of the vaccine to fight disease. So you can push it out beyond the 3 or 4 week window and it should be okay.
Cool. Yeah, that's definitely good to know, and that's pretty much aligns to what I was expecting in that regard. Yeah, because I'm especially thinking, when mixing vaccines that are not of the same technology, like let's say Pfizer or AstraZeneca, I suppose that could potentially have some strange effects. I mean, we don't know. You said it yourself. We don't know. I actually have a little bit of a similar kind of questions about the idea of mixing vaccines. That's not exactly the same, but because here we're talking about mixing vaccines, like having the first shot be one vaccine and the second shot be another. But now the second question would be more about okay, you got your first, you got your 2 shots of whichever vaccine you took, and it's got its efficacy for a certain period. Now, once its efficacy runs out, if you take a different vaccine, I mean, I don't think it would be any difference by definition, but in that kind of situation, is there anything to consider, do you think? Or
yeah, no, I think that's a great question. I will say now that it is more likely than not that we will need a booster at some point in time. And I'm sure that's one of the questions the researchers are asking is, can we do booster tests on different vaccine platforms with people who got either Pfizer, Moderna, Johnson & Johnson? We try them with all the others as boosters to make sure that, yes, it's possible that even if you don't have the vaccine accessible, you can still get that booster shot.
Okay, maybe to ask a follow-up question on that. Now, Now let's say let's take a bit of a look in the future. Let's say we got most of the world. They all got their second shot now, and I don't know what is the future holding for us in terms of COVID. Are we gonna need an biannual new doses of vaccine now, or?
So that's a great question, and the correct answer I'll start with: we don't know yet. But what we can absolutely say is, so this is not the first coronavirus that we've ever been infected by. There have been 7 other coronaviruses that have come into the human population. 6 others. 2 of them were SARS and MERS, which we handled pretty quickly, and it didn't have the global spread that this one does. But there's 4 that are always in the human population, leading to common colds. 20% of your common colds are coronaviruses. And why that is because coronaviruses tend to mutate and evolve and find a way to escape your immune system. And so one of our researchers at Fred Hutch studied this evolutionary trajectory of one of the coronaviruses that is already present in the human population and tried to figure out how it's been evolving over the years. And you see this evolution of that virus. So it's possible we'll see the same thing with SARS-CoV-2. Having said that, there's another researcher at the Hutch who's trying to take a different approach, where he says, let's look at all the coronaviruses, and can I find an antibody or a treatment that can target all of them? Because if it can target all of them, then that suggests that there's a piece of a coronavirus that cannot mutate or evolve, because if it does, then the virus would be dead. So can we target an unchangeable part of the virus with a treatment that will hopefully then give us across the board coronavirus treatment that you would never need to get treated ever again or get vaccinated ever again? So there's a few different ways that this can go, right? Like, we could be getting rid of 20% of all common colds, which not a bad thing in and of itself as well.
Yes, indeed, getting rid of the COVID would be really nice. I even remember, like, last time back in winter 2020 we talked. I think you said, like, the measures we took, like, all over the world to prevent, like, the spread of COVID, like, actually did help, like, basically stop the flu. I think you also said, like, back then, on the topic of like yearly jabs, that, for instance, you've got your yearly flu jab, but that it wasn't as likely for COVID. I think I'm just remembering from memory here, that it wasn't as likely for it to happen for COVID to have, like, for have that booster to be yearly or anything like that. But I mean, at this point, as we've discussed, it looks like a booster probably will be likely, but once again, we don't know exactly. It will really depend on.
I mean, it depends on 2 different factors, right? Like, it depends on. So right now we have data for 6 months on how long people have protection, and we know that people have protection for at least 6 months. We don't know about 9 months. We don't know about a year, because, well, we haven't tested the vaccines that long.
By definition.
By definition, we haven't tested for that long. So once we figure that out, maybe that will shape when we get the boosters. And the second is the variants, right? Like, as the virus faces an immune system that has learned to recognize it, will there be new variants that show up? Will there be new mutations that happen in response to people getting vaccinated, leading to a variant that will be able to escape vaccination? And then we'll need to get a new shot out for it. So there's 2 parts to this. And so one of the ways that we get variants is that there's uncontrolled spread of the virus. And that's why it's really important to bring that down while we're still vaccinating, to reduce that probability that we're getting more and more variants showing up.
Exactly. Flo, have you got anything you want to add to that before?
Let me take a quick look. Yeah, I think in terms of COVID-19, we have we've said pretty much everything. Maybe a quick look at some submitted questions in here. Yeah, so after vaccination, I'm so how far? So I needed to read through the question real quick. So basically, we don't know if we're 100% immune to the virus once we're vaccinated in terms of transmitting it, but in terms of being vaccinated to not vaccinated, how much more likely are these chances of transmitting the virus to someone else?
Yeah, so that is a great question. And we don't know the answer, but we will shortly. So Fred Hutch is also helping lead a trial called COVID-U, where we are working with the US government and with the vaccine makers. We recruited college students on a college campus, half of whom will get the vaccine, half of whom will not. And then they will swab themselves every day. So then we can see whether the people who have the vaccine can transmit the vaccine less well than people who don't have the vaccine. So if I've been vaccinated, am I able to transmit the vaccine as efficiently as somebody who has not been vaccinated? And so then we'll really know whether the vaccine prevents transmission. As of this moment, we don't know. But I will say that real world evidence of what's going on in the UK and Israel does suggest that the vaccine possibly does play a role in reducing transmission as well. Because in Israel for sure, once the elderly populations got infected, you saw a rise in infections among the younger population, but no rise in the elderly population, suggesting that they can't get infected and possibly they don't transmit it as much. But this is all anecdotal. We just have to wait for the test results from the trials that Fred Hutch is running.
Oh yeah, that'd be really nice if. Oh, go ahead.
Well, when exactly can we expect these results?
So yeah, there's a couple of ways. So the trial launched last month. We're hoping that we will have that data by the fall to sort of inform colleges what to do going forward. Because I mean, I'm a parent. I'm glad my daughter goes to daycare. I feel for all the parents whose kids are at home and can't go to school, and they have to have, Wi-Fi split 17 ways so that everybody can go to school, go to do work, and do everything else. I feel for all those groups, and understanding that is really crucial to thinking about how we move forward with getting schools back as well.
Yeah, I think when you think about the COVID pandemic, there's also all the other effects as well. For instance, I don't know if it gets a bit off topic here, but there's the effects. Like for example, on parents, on mental health in general. Actually, I don't know if it's actually a question that relates. That's actually outside of like our COVID-19 section here, but I suppose it kind of relates to that as well. And I think it might especially relate because Fred Hutch also being a cancer research center. With COVID-19, this pandemic being present, I suppose that many people might have misdiagnosis of not just cancer but other illnesses as well. Like, do you think that could be like an actual big risk that could be seen like in the future of people potentially dying because they couldn't have seen they couldn't have been diagnosed early enough because they couldn't get their diagnosis due to the pandemic? Is there evidence of that happening since it started?
There is already evidence of that happening. The number of missed screenings has been due to COVID-19 pandemic has been really high. And I think I saw a paper which suggested that now people are getting diagnosed later. So the cancer has spread further due to the fact that the early screenings and detections were missed. And so that effect we will be seeing for years to come, actually, the missed screenings. But the hope is that some positives have come out of this, like telehealth has improved, hopefully, and talking to your doctor and making sure that everything's okay. It's easier to get access to a doctor to make sure everything's okay. But hopefully, so other options, like, so a Fred Hutch researcher has been really pushing for this test for colorectal cancer. It's called the FIT test. And for colorectal cancer screenings, it just involves mailing something to somebody's house, them taking a sample and mailing it back, and using that for diagnosis. And so changing how we think about how we diagnose people will be really important going forward. So hopefully this will lead to some flexibility and some new ideas and new technologies that can improve screening even remotely and change how we do this. And that is really crucial because that changes access for underserved communities and people of color who don't have time to go to the doctors or don't have as frequent doctor visits. So finding a way to improve equity is really at the forefront of this.
Yeah, that's one good thing that's taken out of it, at least.
Yeah.
Flo, you were about to say something?
Yeah, I just wanted to move on here with the next question, which is we have seen you've talked with us in the past about how the development, how the scientific evidence of HIV vaccine has benefited COVID vaccine. Now, how does it look in reverse? How did COVID vaccine benefit HIV vaccine?
Yeah, that's a great question. So what we spoke about previously was the idea that mRNA vaccines were being trialed for HIV even before COVID came along. So we've been testing these different vaccines, and even the treatments like the Regeneron monoclonal antibodies or the Eli Lilly monoclonal antibodies, they'd been trialed in HIV before. And so that therapy moved over. But the speed at which things were developed for COVID-19 will really have a huge impact on HIV, because HIV changes and mutates far more frequently than COVID-19 does. But the scale to which we were able to make these vaccines, these mRNA vaccines, so quickly, the sequence for the virus landed in somebody's inbox on January 10th. By February, we were already making that mRNA vaccine. So that speed and technology, that adaptability is going to be crucial for somebody with HIV, where the virus is constantly evolving so quickly, and even within populations, that you can update that vaccine really quickly with this mRNA technology. And it's not just HIV. mRNA vaccines could be the future for cancer treatment as well. If you think about it, it could be getting to that world of personalized cancer treatment, where you understand, oh, the surface of this person's cancer has this unique element that is not present anywhere else in their body. Let's make a vaccine to that and train the immune system to recognize that and destroy it. Because all you have to do is print out an mRNA fragment, and now we know how to make it.
So the mRNA can be basically individual to like potentially. Oh, yeah, I can see why they could be really crucial, for cancers potentially. Once it develops even further.
Yeah, and that's the hope, right? Like you can, to get to that next level of personalized therapy that is quicker to make.
That's true. Oh, go ahead.
No. I just if you wanted to kind of go on with that question, I just would have moved on with the next question again.
Oh yeah, because I was just thinking in general. It's interesting to keep an eye on the future of this technology, because we've been mentioning in the case of HIV as well as cancer, and of course obviously COVID-19, because Fred Hutch is quite well known for its cancer treatments, like including immunotherapy. Even wasn't it like one Fred Hutch researcher who won a Nobel Prize, if I'm not mistaken, for their research? Like, could you actually do a little reminder on that?
So there's been 3 Nobel Prize researchers at Fred Hutch.
Oh.
One developed the bone marrow transplant, which is the curative therapy for blood cancers, which led to today's immunotherapies. But the other piece of it is also 2 of the researchers did basic science. So one of them studies how cells grew and divided. And that's been really crucial because the mechanisms that he discovered are often the mechanisms that are changed in cancer. And now we're developing treatments that target those specific mechanisms. So for me, I'm a basic scientist. I believe that it is important to know everything about everything. And so doing our best to understand why things work will really help us to treat when things don't work properly. So the other researcher actually studied the sense of smell. They discovered how smell works, which is super interesting.
Really? When was that? I'd be curious to hear more about that work then.
Yeah, that was work done in, I want to say late 90s. So the thing about Nobel Prizes is they give the award after many years when they found that the technology works. So that work was done, I want to say early 90s, late 80s, early 90s, and the Nobel Prize was given in 2004 for it.
Yeah, that's what I think. I briefly recall, like when I was seeing, indeed, that was around that time period that it was given, but the research itself was in the 90s. Okay.
Yeah.
I feel like I could easily ramble on for very long here, so I feel like Flo needs to stop me before I start asking 1,000 questions about all that.
I'm happy to talk about our neural.
I think this could still very, very well be interested, but so to have some other, you could almost say kind of a random question. So what do you think what are some more undervalued areas in research that don't often get the attention, but that you think still have potential or are still very important, and for whatever reason do they not get the importance they need?
So the big research that is flying under the radar that I am super blown away by is the research on the microbiome. So the microbiome are the bacteria and viruses that live on our bodies and in our bodies. Believe it or not, there are as many cells that are bacteria and viruses on your body and in your body as there are cells that are you. So 50% of you is bacteria and viruses, and 50% of you is you. So you're actually sharing space. And
freeloaders.
They're all, yes and no. They actually help you a lot as well. There's a thought that the microbiome in your gut can break down certain foods and get rid of certain things that would lead to cancer. So having a good gut microbiome can reduce your colon cancer risk, because there are some bacteria that will take all the food that's bad for you or could lead to cancer and just like break it down, and it's never present. And so the contents of your gut microbiome could really affect your cancer risk and your health risk. And your gut is also where your body learns what is friendly and what is unfriendly. So your immune system is really shaped by your microbiome. And so the possibilities are endless with the microbiome.
That definitely makes sense. Jokes aside, it's in the word biome, in the fact that by definition, it's all one, like all these systems they're together, right. And so it makes sense that as such, they can be like very important, I suppose, then, as you say, in this kind of things.
Can I describe it? Examples on this actually real quick before I go on?
Yes, of course.
one is that we have a researcher who's discovered that when newborns are born, so this is all done in preclinical systems. When newborns are born, they're born with a microbiome. And then the mother passes through the breast milk antibodies that recognize the microbiome. And if they don't pass those antibodies, something happens with the newborn's immune system, and those newborns don't grow as well or as effectively as people who get antibodies from the mother's breast milk.
Okay.
Those newborns grow far better than those that don't get it. So like even newborn development could be tied to your microbiome and the dance between the microbiome and your immune system. And the other story, which is even cooler, is so we haven't known about the microbiome because we didn't have the technology. Like when I started my undergrad was when we just learned how to sequence DNA. I was just talking to my old advisor how we'd send something for sequencing and it would be 2 weeks of a wait before we knew what the genetic sequence was. Now we can sequence whole genomes in hours. And that technology has evolved. And now we're starting to detect more sequences that are not human. And back in the day, we would look at all the genomes of cancers and be like, okay, this is not a human sequence. We'll throw it out. We'll throw this out. But now when you add those back in, you're starting to see that a large number of solid tumors have a microbe associated with it. So a large number of our cancers may be driven by a microbe. And so the cancer treatment may not be treating the cancer, but be treating with antibiotics to kill that microbe, which will totally revolutionize how we treat cancer going forward. It'll be treated as like an infection rather than something worse.
Oh,
Okay.
now I understand why you're fascinated by that field.
Yeah.
It's very interesting. Yeah.
Wow. Do you want to say something, Floa?
I'm just looking at the doc. I don't really have anything to say.
Yeah.
Maybe you still have something to say, Pika?
So, yes, indeed. I'm not sure how much on, like, it's kind of relating back to COVID-19. So I guess we're just gonna again whisk back a bit to that
Sure.
topic a bit. I suppose it's also related kind of to the way data works and the way we interpret data. As we've kind of mentioned already, the current situation in India is, it's absolutely awful right now. And we've talked a lot about the variants that are in India. And something, as you were yourself saying, before we started the interview, therefore, what about how could we misinterpret the link between the variant and just the virus spreading because, not of the population, I'm not sure what would be the best term of it, but just to, outside of, of the variant itself, it could very well be spreading not because of that, So when looking at data about, of course, I'm going to say about COVID-19, but it could be about anything related to that. What do you think are the things we need to be careful about when reading such data?
Oh, absolutely. I think this is a great question. I think, every time a variant is discovered somewhere, everybody is very quick to attribute a lot of the spread to a variant. But there's more to it than that. It's important to really have good genetic sequencing in every country to look at all the cases. And based on what we've seen, a Fred Hutch researcher, Trevor Bedford, whose website NextStrain tracks viruses as they move across the world, and he's been tracking all these variants. He basically looked at all the available sequence data. So caveat is available sequence data. And he found that there were 3 or 4 variants that were growing at exactly the same rate. There wasn't one overtaking the other, which suggests that it's growing all through the country. It's not being driven by one variant per se. And so it's important to really dig beneath the data and say, okay, the cases are going up. Yes, there's a variant. Is that variant driving the rises or not? But for that, we need good data and good sequencing, which in India, we don't really have yet, but we definitely need to get for sure. And
Yeah.
Yeah, go ahead.
No, I was just going to comment, right now, I suppose what India needs the most right now is support for the healthcare system right now, which must be saying that it's overwhelmed is an understatement, because right now it's, yeah.
Yeah. I mean, I'll add one more thing, right? Like, it's not always the variance, because there was a new paper published by Trevor and his group that looked at Washington State. So admittedly small sample size, but a decent enough sample size. And in Washington State, some of you may remember a year ago, we were talking about this D614G mutant and this variant that was showing up that was more transmissible than the original. And what Trevor, they did an analysis and they found that the thing that was really spreading the virus was people's behavior. People going out, meeting with one another, not following lockdowns. Whenever we were in lockdown, the levels fell down regardless of variant or not variant. And there is a human agency that we are not willing to accept yet. And I think we need to do that. Like in India, there needs to be a lockdown. There needs to be a full-scale lockdown and nobody going out. Mask wearing mandated, because otherwise those are some of the tools we have, and we have to use every tool at our disposal with what we have in India.
Yeah, human agency, that's the thing indeed that's very important.
Yeah. I mean, we're all looking for a silver bullet, right? But for a lot of things, there isn't one. And for COVID-19, it's just, there's a lot of imperfect factors that you just have to layer on. So I heard this great analogy. It's like Swiss cheese. Stopping COVID-19 is like stacking up lots and lots of layers of Swiss cheese. There's holes in all of them, and they're all slightly imperfect. But if you stack together enough layers, you can stop the spread, because the holes won't line up in all the Swiss cheeses.
I like that analogy.
Yeah, that's an interesting analogy.
Okay.
Yeah. Delicious too.
Just don't eat up all the plan, or it will all go back to horror then.
Just make sure you stick to it, and
just have the diligence to not eat the plan. There we go. Were you going to say something, Flora?
No. Just keep going.
When thinking about the idea of data, a lot of this just kind of reminds me also, likewise, on the topic of the vaccines. Because one, of course, one of the big subjects of controversy in the past couple of months has been, like, one of the most famous examples is, for instance, the blood clot with AstraZeneca and that kind of thing. And what, I keep wondering about is, okay, for instance, you've got that many cases, of blood clots happening compared to that many people that actually got vaccinated with AstraZeneca. So I'm just wondering, is it then a justified interpretation of the data that caused the controversy, or is there perhaps a hint of panic there? What would be your opinion on that?
Yeah, I'll say this, right? Like, it is, the pauses for AstraZeneca and Johnson & Johnson were essential, and they needed to happen. Because what happens with these vaccine trials, if you think about how a trial is constructed, it's 30,000 people that are divided into two groups, half get a vaccine, half don't. So effectively, only about 15,000 people are getting the vaccine. 15,000 is not 10 million. It's not 15 million. It's not 100. There are unique people everywhere. And so when you go from vaccinating 15,000 to 15 million, you'll find people with rare symptoms or things that could lead to some side effect. It's important at that moment to pause and say, what is common amongst the people that are having these side effects? And can that change how we recommend the vaccine, or who we recommend the vaccine to? and how long we monitor the people who are getting the vaccine. And it's important as we're rolling these out to get that information to learn. And yes, the numbers may seem small, like Johnson & Johnson, it was what, 1 in a million. But those 1 in a million, it was a smaller percentage of women between 18 and 39 that were getting the shot. And so they needed to be watched for a longer period. So it's important to change the guidelines as we learn these things.
Okay, yeah, this actually does make a lot of sense to me indeed, because it's not just the numbers when it comes to statistics and data. There's also the medical data about people and other individuals. And as you said yourself, by definition, when vaccines are in trials, especially when done so quickly, you can't test the vaccines on every single type of people. Right. So in that sense, the idea of pausing to determine exactly what is the problem, what aspect of people is causing those, like in those kind of examples of blood clots or whatnot. Okay, yeah, that sounds that seems fair to me. Okay,
and I would love it if it was reported as such, right? Like I would love it if it's like everything's okay. We're just checking to make sure who we change advice to. This is part of the scientific process. Science is messy. You have a hypothesis, you test it, you get some results, you revise your hypothesis. And that's, it's just being done in real time with everybody watching.
I suppose I suppose the media making a panic out of it, that can create some issues. Because, putting it that way, it's, I guess, trial and error, I suppose.
Right.
The scientific method.
Yeah, I think it's in general, people want, I guess people just want this one definite truth from the science. And if they suddenly realize that there is not this one definite truth, or that what we thought was the one definite truth, is it not, then it does kind of cause an uproar, maybe to say.
And exceptions don't prove the rule, but it's important to know what the exceptions are and why they exist and solve for that problem.
I think, speaking about that, I think that really highlights the importance of knowing how to read data and how to interpret it. Because, otherwise that can easily create a mess when that's not the case. I think I saw on the Fred Hutch website, actually, there was like a division on data science, or wait, let me actually see.
Yeah, we have a division of biostatistics. We have a division of data. We have a data science group as well, computational biology. We do a lot with data. And, as technology gets better, every experiment yields exponentially more data. So there's a lot of data going around. So if anybody here or anybody listening is a good programmer, a lot of biology is becoming programming, and we need programmers to understand the giant data sets that we're getting.
Yeah, you've got a whole field there, like a whole profession. Yeah, with so much more data to interpret, so much more data to process. Wow.
I mean, there was a really cool study done by a group at Fred Hutch and University of Washington that basically was able to try and get to the question of like how every cell in a developing creature becomes the final cell. Like how the programming of a cell changes as the animal develops to know exactly what each one's outcome is. And so all we're trying to do is understand the programming of trillions of cells at the same time, which is insane.
Yeah.
Insanity.
Yeah.
We've talked a lot about trials, but maybe you could go into deeper details how some of these trials look like. What exactly are you doing?
Yeah. And let me focus on the COVID-19 vaccine trials, because that's actually been a source of hesitancy for a lot of people. as to whether or not they should get the vaccine. So I just want to address it here. So what happens with these trials is there's multiple phases. So there's a phase 1, a phase 2, a phase 3. So the first one, you're just looking for a dose that works. Like you test a range of doses on a group of people to find the optimal dose. In phase 2, you're doing safety and efficacy. You test more people to see how safe it is at that dose. And then you go to the big trial, which is the phase 3, which is the one we're talking about. And so that one, it recruits either 30,000 people or 45,000, depending on the thing. And the groups are split into at least 2 different subgroups, if not more. And half of them get the vaccine, half of them get a control, which is called a placebo. And they're monitored for illness. And basically, once a certain number of people get ill or report feeling ill, that's when they look and see, of the people who got ill, how many got the vaccine, how many got the placebo. And based on that, that's where we get the efficacy numbers from. And so what happened in this instance is that once phase 1 was launched, there was a ton of investment to launch phase 2 even before phase 1 was completed, in the hope that it would be successful. And then phase 3 was launched way before phase 2 was completed. So normally it's you wait for one phase to end, then you get some funding, and then you start phase 2, then you analyze that, get some more funding, start phase 3. This time it all got smushed together, and a lot of these were done at cost. Like the government assumed a giant amount of risk and invested a lot of money in trials that could potentially fail, just so that everything was ready to go faster and we could get these vaccines quicker.
Yeah, you can see why. It's the speed that sometimes can create that doubt. Then once again, especially like when it's not even done.
Yeah, safety was not compromised. It was just risk was assumed. And with those 30,000 people, they're monitored every month, every 2 months, every 6 months, every year. So those people are still being monitored for up to 2.5 years to look 2 years to look for any adverse effects at all, and to look for protection from the vaccine. So they will continue to be monitored. So the trials are still ongoing for them.
I see.
It's interesting to know.
Yeah.
To get this this kind of behind the scenes look.
Yeah. And again, the trials were meant to look for illness, symptomatic illness. They weren't meant to look for anything else. That's why this new trial, which is looking at college students to see whether they can transmit the virus, had to be a fully different study that was looking into this.
I see. Okay. It's just so why all of that, because I'm just kind of getting lost in my train of thought here, because there's obviously a lot to process here, but I think there's definitely a lot to take away from it, and that's what's good with these talks, let's say.
I mean, and I'll add one more thing that you are likely to start seeing in the news a lot more. And it's this term called correlates of protection. And so what that is, we did these trials. They lasted for 6 to 8 months, and we waited for people to get sick.
Okay.
Do we have the luxury, if COVID mutates and changes and we need another vaccine, to wait 6 to 8 months to run this full trial? Not really. And so what correlates of protection are is, now that we have all these trials that we've run, we can look at the people who did get sick after vaccination and those who didn't, and say, what elements of your immune system were present in the ones that didn't get sick and were not present in the people who did get sick. And so instead of waiting for people to get sick, we can just look for certain markers in your blood that says your immune system is active and it is likely to keep you from getting sick. And that way, once you give people the vaccine, these test vaccines, within a month or two, you can know, okay, here's the spread of these certain factors in the immune system that we are seeing. So that vaccine should be safe enough and prevent against disease as well.
Okay, so it's basically this extra data that's been obtained from these previous trials
Exactly.
can then help then in it also having the development of potential new versions of the vaccine in the case that new ones like need to be made.
Or even getting data on the vaccines in younger children, right? Like, you don't need to test all of them. You're just like, wait, okay, we look at the blood for a few months. Okay, this lines up with what we saw in adults. We're okay. We can go forward with this vaccine.
Okay.
Now, this is, I don't know how to best formulate this. I mean, there's always this kind of risk with these trials going on, I guess. But like, how much willing are, I don't know if this is kind of how much risk are you willing to go for? Maybe.
So that's a good question. And I think it depends on the trials, right? Like, we ask for volunteers and people drop out of these trials often, once presented with the information. But there's been an overwhelming response. Normally, to run a trial this big takes a long time to get these people that are willing to volunteer for these trials. Here, people have been willingly volunteering in giant amounts that we were able to fill up these trials so quickly. And it's not a risk to sign up for these vaccine trials because we've tested them for safety. We're just now trying to see whether they protect you. And that's it, right? And all the people who get the vaccine are coached and they're told, here's what you have to do to prevent illness. Please follow these guidelines. Please don't do this. Please don't do that. Please wear a mask. And then you just monitor if you're assuming people are on best behavior. It's not like we're giving them a vaccine and saying, now we're going to inject you in the face with COVID. There are some trials like that. Those are called challenge trials. And there's a huge question about the ethics of that. They work if you have a treatment for that. So there are challenge trials that are done for malaria all the time because we have a treatment that can stop you from getting sick and dying from malaria. But for COVID, we didn't have that. So these trials for COVID vaccines are not putting people at any more risk, really, than getting the disease itself. But I see Pika has a follow-up on challenge trials, probably.
Yeah, I was just wondering, so what would be the quote-unquote benefit of doing challenge trials compared to, let's say, how you're describing the current COVID-19 trials? Like, why?
Yeah. So that's a good question. So with these current trials, we're just waiting for people to get sick. We're trying to see how long it takes for people to get sick. And once you reach a certain threshold. With the challenge trials, you get the vaccine, you wait 2 weeks, you expose people either with the vaccine or without the vaccine to the virus, and you just count how many people get. So you have the data in 3 weeks versus 6 months.
Okay,
maybe. So you said you wait for people. You wait for people to get sick, but what are you doing? What are you doing then? Maybe the question.
At that point, we're just everybody gets a weekly check-in of how are you feeling. You get a symptom screener, and that's it. It's that's all we're asking. Are you feeling any of these symptoms? And the second they log, okay, we're feeling these symptoms. You make a check mark and you say, what are the symptoms, et cetera, et cetera. They're getting advised on treatments, et cetera, all during the course of that. And so once in these trials you hit, say, I think it was like 170 something illnesses, that's when you could look at the data in an unblind fashion. So this is all done blinded. So the person doesn't know whether they've got the vaccine or the placebo. The person administering the drug doesn't know if they've given the vaccine or the placebo. Nobody knows. And once you hit that number of people who are infected, then you can say, okay, who got what? And then you count how many of the people who got sick were with the vaccine and how many were with the placebo. And usually it's been like 10 with the vaccine out of 15,000 versus 150 without the vaccine have gotten illness. And that's how you figure out these numbers.
Okay, I see. Wow.
There's a lot to take in.
Sorry,
It's,
science is complicated.
Yeah, but still, it's not that it's necessarily complicated, but it's really interesting to just hear how all of this process works, like all of this. You I don't know, when you usually just watch, I don't know, TV talk shows or like that, you don't get these kind of information.
I mean, I think it should be more publicly accessible. That's my personal opinion. The more science does to be accessible, the less mistrust of science we get. This is how we do it. For better or for worse, this is the thought that went into it. Here's what we still know. Here's what we do know, and here's what we don't know. And that's important to say that there are things that we don't know.
Yeah, I think what's key to it is the honesty, that comes with it. I mean, it's not just about not just in science, but in general. It's always an important thing to admit that you don't know everything, and part of science is is basically the same, because you're constantly doing that research. And I mean, it's not for nothing that a lot of the answers you gave, when we asked the question, was we don't know yet.
Yeah.
And a lot of it because, simply put, we're there's still so much yet to learn about COVID and how it's going to be, and how the vaccine are going to impact things in the future, because we're still just barely in the beginning, and it's impossible to predict with 100% accuracy what's going to happen. You just can't do that. And
yeah,
I think that,
as you just said, that it's all just predictions, and predictions are never really 100% accurate.
But you can make educated guesses based on priors and based on currents, and that's all you're trying to do, is make the best possible guess you can on the information you have available.
I actually think all that really does a really good summary of this whole talk we've just had. Like, this whole talk about educated guesses and predictions and not knowing, and basically getting into the unknown with all of that. I think personally, this does for a nice closing message.
Oh yeah.
Just to we admit we don't know. It looks like the world doesn't know everything, and the world is still searching for answers regarding COVID and its impact. But progress is being made. That's what we can see here.
And I'll say this much, right? Like, I left the bench 3.5 years ago. I don't miss having my experiments fail all the time, which they did. I wasn't great at science. But I do miss the thrill of when you discover something and believing you're the only person in the world that knows it. Because you are. You've asked a question that nobody's asked before. And there is something that comes with that. It is really fascinating.
Maybe if you allow me a kind of personal question, what exactly has moved you to become this scientific advisor instead of leaving the scientist behind, basically?
Yeah, that's a good question. So there's a couple of different factors. I always wanted to be a teacher. I, for years, have wanted to go teach at a university somewhere rather than continue research. I love the research. I know what I'm good at. I know what I'm not good at. And I wasn't great at formulating hypotheses that would get funded. And I wasn't great at getting that funding. But I love talking about science and I love teaching it. And so in this role, it's sort of perfect for me. I am teaching effectively.
Yes, indeed. I think just understanding yourself and where your areas of strength and weaknesses are is quite an important part of like just everything.
Yeah.
I think just all in our own ways, we're all always searching for those, all those ways.
And I got lucky. I fell into something that I enjoy and I'm good at. And, yes, I had to shape my own career a little bit, but you adapt, but stick to your strengths.
That's nice to hear.
Yeah.
Where can people keep up with all that is happening in the science?
So you can always go to fredhutch.org to keep up with some of the research that we're putting out there. You can even follow some of our researchers on Twitter, or Fred Hutch on Twitter and Instagram. And even on Instagram, you'll see me doing some stupid things in the name of science to talk about science as well. But so Fred Hutch on Twitter, Instagram, LinkedIn, whatever your choice of social media. And on Twitter especially, that is the more scientific side. So we do retweet a lot of the science that is going on at the Hutch and with our collaborators.
I actually have like kind of a follow-up question to that, which is really, really similar but kind of different.
Yep.
Which is for someone, let's say, to want to become more scientifically informed in general, what would be, the best sort of steps that someone can take to generally keep up with science in the best informed ways, if you will? Does that make sense to you?
Yeah, no, it does. And that's a great question, because I'm actually not sure I have the perfect answer for it. Just depends on what your area of interest is, and it can change based on what the area of interest is. I mean, I know for me personally, I read all the work that happens at Fred Hutch. I listen to a bunch of talks. So actually, even going to Fred Hutch's YouTube channel, I do put up a lot of our scientific talks that you're welcome to go listen to, including one on the microbiome that I put up maybe 2 months ago. So that is worth a listen to. I also read certain publications like Vox News has a very good science writer as well. And Stat also has a really good science section as well. So a couple of the publications that do a better scientific writing I read as well. The Atlantic as well in the US also has a really good science writer as well.
Okay, yeah, that's perfect to know, because sometimes it's such a vast world, it's kind of hard to know, if someone's just got an interest. Of course, now it's really about so you're just curious and, okay, where do you even start if you're curious to learn about something? Well, exactly.
Yeah.
So what is it that you do at the Hutch? Where is the money going? Why should the people donate towards Bingothon?
Yeah, I mean, the Hutch is a comprehensive cancer research center where we're looking for treatments and cures for cancer and other diseases that are affecting people around the world, like HIV, malaria, COVID-19. And a large amount of our funding comes from the government, which is great. Like, it provides the backbone of our research funding. But the way government funding is structured is it likes ideas that are already semi-proven, and then they want to take it to completion. But those like crazy ideas, those little brilliant seeds, those aren't funded by government money. That comes from individual donors and from philanthropy, from people at Bingothon. Like, you are funding the next big idea, the next thing that could cure COVID-19 or HIV, one of those things. Like, those brilliant seeds of ideas, that's what this is funding.
I think we can soon slowly start to get towards the end. Just, is there any final kind of things any of you want to say regarding this whole topic, as a whole? Just anything you have in mind.
Well, thanks for Guppy for talking with us again. this was a very interesting talk. Lots of information, and yeah.
And I'll say, I am honored to be talking with all of you. And honestly, like, if you find me on Discord and you have any questions, shoot them my way. I don't think we should be restricted to just Bingothons to talk about any questions you may have. You can definitely send me a message on Discord, and I'm happy to answer those questions as well.
Yeah, just find Guppy in the Bingothon Discord and send him a message.
Yep.
All right. Yes, exactly. I mean, you indeed, Guppy is right here in our server, so sending a message is very easy. So indeed, I also have to say my thanks to Guppy for taking time here. For our talk / interview / whatever you want to call it. I also want to thank whoever made it this far into the video, because we kind of rambled occasionally. We talked about many things. It's been a wide variety. At Bingothon Summer 2021, our intention, was to found, not just for COVID, but also for all of what Fred Hutch does. And I really hope this video helped you understand more about what it is that Fred Hutch does, and that, well, first off, that you enjoyed the event, that whether you were able to give or not any donations, that at least just that extra knowledge could be useful for you. So yeah, thanks to you. And yeah, we hope you'll be enjoying our next events. There we go.
Bye, everybody. Thank you.
All right.
Yeah.
Thank you, everyone, and bye.